Today, we're diving into a topic that bridges ancient wisdom with modern science: ginsenosides. These powerful compounds, derived from the revered ginseng plant, are gaining significant attention for their diverse biological functions and their promising applications in veterinary medicine. Many of us know ginseng as a popular supplement for humans, especially in Asian cultures. But its active ingredients, ginsenosides, are proving to be much more than just a human health fad. They're a class of natural steroid glycosides and triterpenoid saponins with a dammarine-type structure found primarily in plants like Panax ginseng myrrh and Panax notoginseng. Now, researchers are systematically investigating their potential to improve animal health, boost productivity, and offer new therapeutic avenues in livestock, poultry, aquaculture, and even companion animals like dogs. While they offer exciting possibilities, it's also important to acknowledge that ginsenosides face challenges, including poor water solubility, short half-life, and low bioavailability in animals. But the benefits could be substantial. Let's break down their incredible biological functions and then look at specific applications across different animal species. The multifaceted biological functions of ginsenosides. Ginsenosides don't just do one thing, they exert multifaceted regulatory effects on animals, primarily by modulating critical signaling pathways and interacting with cellular receptors. Think of them as tiny biological master keys, unlocking various beneficial responses. Here are some of their key functions. Antioxidant powerhouses. Animals in intensive farming, facing stressors like weaning, vaccination, or temperature changes, often accumulate reactive oxygen species, RS, leading to oxidative stress and cellular damage. Ginsenosides come to the rescue by scavenging free radicals, inhibiting nitric oxide, NO, production, inducing the expression of antioxidant enzymes like catalase, CAT, and superoxide dismutase, SOD, and reducing harmful lipid peroxidation. A critical mechanism involves activating the NRF2 signaling pathway. This pathway is like the cell's internal alarm system for oxidative stress. Ginsenosides promote NRF2's movement into the cell nucleus, where it binds to the antioxidant response element, R. This binding then upregulates the expression of other antioxidant enzymes such as heme oxygenase 1, HO1, and DPH, quinone oxidoreductase 1, and QO1, and glutamate cysteine ligase catalytic subunit, GCLC, significantly enhancing the cell's ability to combat oxidative damage. For example, ginsenoside RG1 has been shown to protect H9C2 cells from hypoxia/reoxygenation injury by activating the NRF2/HO1 pathway. Anti-inflammatory agents. Inflammation is a natural immune response to infection. Ginsenosides suppress pro-inflammatory cytokines like TNF-alpha, IL-1 beta, and IL-6 by inhibiting the degradation of IB and the nuclear translocation of NF-kappa B. NF-kappa B is a central transcription factor in inflammatory responses, so blocking its activity is key. They can also interfere with the toll-like receptor 4, TLR4, pathway, which is often involved in triggering inflammatory responses. Ginsenoside RB1, for instance, has been shown to protect mice from LPS-induced liver damage by inhibiting this pathway. Similarly, RG1 can attenuate inflammation in cardiomyocytes. Beyond these, ginsenosides modulate other crucial signaling pathways like JNK, ERK, and P38, further reducing inflammation and cellular stress. Immune regulation boosters. Ginsenosides play a significant role in enhancing both humoral and cellular immunity. They can promote the production of serum immunoglobulins like IgG and IgM in mice. They're also being explored as vaccine adjuvants. Studies have shown that ginseng stem and leaf saponins, GSLS, can enhance the immune efficacy of infectious buccal disease virus, IBDV, and Mucosal disease virus, NDV, vaccines in chickens. Ginsenoside has been found to enhance the immune response to inactivated rabies virus vaccine in mice, and RG1 showed adjuvant properties in stimulating immune responses against hepatitis B surface antigen. Stress alleviators. Stress in farm animals, whether from weaning, heat, or immune challenges, often leads to cellular oxidative stress. Ginsenosides, as we've discussed, mitigate this by activating antioxidant pathways. They also regulate the hypothalamic, pituitary, adrenal, HPA, axis, which is central to the body's stress response. For example, ginsenoside RG3 can reduce serum adrenocorticotropic hormone, ACTH, levels in broiler chickens under immune stress. This suggests ginsenosides could help animals better cope with various environmental and physiological stressors. With such a wide range of biological activities, it's no wonder that ginsenosides are being eyed for numerous applications in animal production. Let's explore how they're being utilized across different sectors of veterinary medicine. Applications in animal production. Ginsenosides are typically administered as feed additives or oral supplements. Their impact is often species-specific, but the underlying benefits of immune regulation, antioxidant effects, and anti-inflammation are consistent across the board. In the swine industry, ginsenosides show great promise for improving gut health in pigs. They can modulate gut microbiota composition, strengthen intestinal barrier integrity by upregulating tight junction proteins like ZO1 and aclidin, and reduce the number of harmful bacteria like Escherichia coli in feces. They also boost systemic and mucosal immunity, elevating serum IgG/IgA levels and activating key immune pathways. This has implications for improving vaccine efficacy against common swine pathogens like PRRSV, porcine reproductive and respiratory syndrome virus, and PEDV, porcine epidemic diarrhea virus. Ginsenoside RB1, for instance, has demonstrated antiviral effects against PRRSV. For breeding, ginsenoside RG1 has been shown to improve embryo quality by increasing glucose uptake and reducing apoptosis of embryonic cells under oxidative stress conditions, suggesting its potential to enhance the reproductive performance of sows. In poultry, adding ginsenosides to broiler diets can significantly improve survival rates, feed utilization, average daily gain, ADG, and reduce the feed conversion ratio, FCR. Studies suggest an optimal dose around 15 mg per kilogram for general ginsenosides or 300 mg per kilogram for RG1. For laying hens, ginsenosides can improve egg quality, increasing egg white weight, eggshell hardness, and yolk content, leading to better egg production performance. This may be due to their stimulating effect on oocyte meiosis and proliferation. In ruminants, while research is still emerging, ginsenosides appear to improve growth performance and meat quality in cattle. Adding 1% ginsenosides to diets has shown positive results, potentially by enhancing rumen fermentation and protein utilization. They can also help alleviate mastitis caused by bacterial infection, with RG1 and RG3 enhancing immune responses and inhibiting bacterial signaling pathways, even competitively binding to TLR4 to exert anti-inflammatory effects in the mammary gland. Ginsenosides can also increase the relative abundance of beneficial bacteria in rumen microorganisms. In aquaculture, the aquatic sector is also seeing benefits, with ginsenosides improving growth performance and feed utilization in fish, including weight gain rate, WGR, feed efficiency ratio, FER, and protein deposition rate, PDR. Fermented ginseng extracts can enhance the antioxidant capacity and immune-related gene expression in species like Carassius auratus by increasing enzymes like GSHPX, SOD, and CAT, and anti-inflammatory factors like IL-10 and TGF-beta. Ginsenosides can also regulate lipid metabolism, promoting growth and significantly reducing total cholesterol and triglyceride levels, as well as liver fat deposition in fish like Silurus asotus. These applications clearly demonstrate the vast potential of ginsenosides. However, like any therapeutic compound, understanding their metabolism and safety profile is paramount. Metabolism and safety considerations. The scientific community is diligently studying how ginsenosides are processed in the body and what their potential side effects might be. Metabolism of 20S, ginsenoside RH2. RH2, a trace constituent of red ginseng, is extensively metabolized oxidatively. This process primarily involves the enzyme CYP3A4 in liver microsomes across various species, including humans, monkeys, dogs, rats, and mice. Interestingly, the metabolism generates reactive epoxide metabolites in both humans and animals. However, a GSH conjugate, M6, a marker of potential reactive metabolite detoxification, was detected only in cynomolgus monkey liver microsomal incubations, not in humans, dogs, rats, or mice. This finding is crucial, as it suggests that cynomolgus monkeys might be a more relevant non-rodent safety species than dogs for evaluating RH2, potentially better covering human metabolites and preventing an underestimation of its bioactivation potential in humans. Studies have also shown moderate oral bioavailability for RH2, 28% in Wistar rats and 55% in beagles. Toxicity of 20S, ginsenoside RG3. A comprehensive 26-week oral toxicity study in Beagle dogs investigated RG3 at doses of 0, 7, 20, and 60 milligrams per kilogram. The study found no evidence of RG3 toxicity in clinical indications, body weight, food intake, ophthalmoscopy, electrocardiogram, urinalysis, hematology, or serum biochemistry at doses up to 20 milligrams per kilogram. Any minor, transient changes in these parameters were not dose-dependent and were considered within normal ranges for the study facility. However, at the highest dose of 60 milligrams per kilogram, the kidney relative weight of animals was significantly elevated compared to control animals, 5.15 plus or minus 0.88 per thousand versus 4.11 plus or minus 0.59 per thousand. This effect was reversed after a 4-week recovery period. The researchers suggest this change might be related to the kidney's role in the metabolic processing and excretion of RG3, as it is primarily eliminated via urine. Based on these results, the No Observed Adverse Effect Level, NOEL, for orally administered 20s Ginsenoside RG3 in dogs is 20 milligrams per kilogram. Previous acute oral toxicity studies in rats and mice established LD50 values of 800 milligrams per kilogram and 1,600 milligrams per kilogram, respectively, and a 26-week NOEL of 180 milligrams per kilogram in rats. Pharmacokinetics of Panax, notoginseng saponins, and 20s protopanaxatriol, PPT, in dogs. When Panax notoginseng saponins, specifically notoginsenoside R1, Ginsenoside RG1, and Ginsenoside Re, are administered orally to Beagle dogs, they are absorbed and metabolized into their saponin, 20s protopanaxatriol, PPT. PPT is a well-known end metabolite with significant bioactivity. The time to reach maximum plasma concentration, Tmax, for PPT is longer than for its precursor saponins, Re, RG1, R1, indicating that this metabolic conversion occurs after initial absorption. While PPT has bioactivity, its maximum concentration, Cmax, in plasma might not be higher than the parent saponins, possibly due to rapid absorption and elimination or further biotransformation into other metabolites. These detailed studies provide a foundation for safe and effective use. However, the journey to fully integrate ginsenosides into veterinary practice is still ongoing. Challenges and future directions. While the evidence is compelling, there are still areas that require further exploration. Unclear mechanisms. The specific mechanisms of action for many individual ginsenosides remain to be fully elucidated. Understanding these in detail will allow for more targeted and effective applications. Dosage optimization. Significant differences in the effects and the appropriate dosages across different animal species are not yet clearly explored. This necessitates more tailored research for each species and production system. Cost effectiveness. The extraction process of ginsenosides is still complex, leading to high manufacturing costs. Optimizing these processes will be key to making ginsenosides a viable and sustainable feed additive option. Human extrapolation, as seen with RH2, in vitro data can potentially underestimate the bioactivation potential in humans, highlighting the need for careful in vivo studies and appropriate animal models. Targeted validation. Specific applications in ruminant and swine models, while promising, still require targeted validation studies. Ginsenosides represent a fascinating intersection of traditional medicine and modern pharmacological research. Their ability to act as antioxidants, anti-inflammatories, immune modulators, and stress reducers positions them as a premium botanical resource for enhancing animal health and productivity. From improving gut health and reproductive performance in pigs, to boosting growth and egg quality in poultry, alleviating mastitis in ruminants, and even enhancing growth and lipid metabolism in aquaculture, the potential benefits are vast. And let's not forget their therapeutic effects on canine inflammatory bowel disease, demonstrating their diverse application even in companion animals. As research continues to unravel their complex mechanisms and optimize their application, ginsenosides could play a vital role in developing new, natural, and green feed additives, contributing to a healthier and more sustainable future in animal husbandry and veterinary medicine. That's all for this episode from the VetMed Digest, keeping minds inspired, hearts light, and tails wagging.